Hi everyone, this is Glenda Ganzon and welcome to my Human Anatomy and Physiology class. And for today's video, I'm going to be discussing the embryologic perspective of the nervous system and the different disorders of the nervous system. So stay tuned. The nervous system is the major controlling, regulatory, and communicating system in the body. It is the center of all mental activity, including thought, learning, and memory. Together with the endocrine system, the nervous system is responsible for regulating and maintaining homeostasis. Through its receptors, the nervous system keeps us in touch with our environment, both external and internal. Like other systems in the body, the nervous system is composed of organs, principally the brain, spinal cord, nerves, and the ganglia. This, in turn, consists of various tissues including nerve, blood, and connective tissue. Together, these carry out the complex activities of the nervous system. There are various activities of the nervous system that can be grouped together as three general overlapping functions, and these are the sensory, integrative, and motor. Millions of sensory receptors detect changes called stimuli, which occurs inside and outside the body, and they monitor such things as temperature, light, and sound from the external environment. Inside the body, the internal environment, receptor detects various pressure, pH, carbon dioxide concentration, and the levels of various electrolytes and all of these are gathered information which is called sensory input. Sensory input is converted into electrical signals called nerve impulses that are transmitted to the brain. There are signals that are brought together to create sensation, to produce thoughts, or add to memory. Decisions are made each moment based on the sensory input. This is the integration. And based on the sensory input and integration, the nervous system responds by sending signals to muscles, causing them to contract, or to glands, causing them to produce secretions. And muscles and glands are called effectors because they cause an effect in response to direction from nervous system, and this is the motor output or motor functions. The brain is a complex organ composed of gray parts and white matter, which can be hard to distinguish. Starting from the embryologic perspective allows you to understand more easily how the parts relate to each other. The embryonic nervous system begins as a very simple structure, essentially just a straight line, which then gets increasingly complex. Looking at the development of the nervous system with a couple of early snapshots makes it easier to understand the whole complex system, and many structures that appear to be adjacent in the adult brain are not connected, and the connection that exists may seem arbitrary, but there is an underlying order to the system that comes from how different parts develop. And by following the developmental pattern, it is possible to learn what the major regions of the nervous systems are. To start with, a sperm cell and an egg cell fuse to become a fertilized egg. The fertilized egg cell or zygote starts dividing to generate the egg cells that make up an entire organism. And after 16 days of fertilization, the developing embryo cell belong to one of three germ layers that give rise to the different tissues in the body. And the endoderm or inner tissue is responsible for generating the lining tissues of various spaces within the body, such as the mucosae of the digestive and respiratory systems. And the mesoderm or middle tissue gives rise to most of the muscle and connective tissue. And finally, the ectoderm or the outer tissue develops into the integumentary system, or what we call as the skin and the nervous system. It is probably not difficult to see that the outer tissue of the embryo becomes the outer covering of the body, but how it is responsible for the nervous system. As the embryo develops, a portion of the ectoderm differentiates into a specialized region of neuroectoderm, which is the precursor for the tissue of the nervous system. So molecular signals induce cells in this region to differentiate into neuroepithelium, forming a neural plate, and the cells then begin to change shape, causing the tissue to buckle and fold inward. A neural groove forms, visible as a line along the dorsal surface of the embryo. The ridge-like edge on either side of the neural groove is referred to as the neural fold. As the neural folds come together and converge, the underlying structure forms into a tube just beneath the ectoderm called the neural tube. Cells from the neural folds then separate from the ectoderm to form a cluster of cells referred to as the neural crest, which runs lateral to the neural tube. The neural crest migrates away from the nascent of embryonic central nervous system, or CNS, that will form along the neural groove and develops into several parts of the peripheral nervous system, or PNS, including the enteric nervous tissue. So many tissues that are not part of the nervous system also arise from the neural crest, such as craniofacial cartilage and bone and melanocytes. And at this point, the early nervous system is a simple hollow tube. It runs from the anterior end of the embryo to the posterior end. Beginning at the 25 days, the anterior end develops into the brain and the posterior portion becomes the spinal cord. This is the most basic arrangement of tissue in the nervous system, and it gives rise to the more complex structures by the 4th week of development. As the anterior end of the neural tube starts to develop into the brain, it undergoes a couple of enlargements. The result is the production of sac-like vesicles. And similar to a child's balloon animal, the long, straight neural tube begins to take a new shape. And there are 3 vesicles formed at the first stage, which are called primary vesicles. And these vesicles are given names that are based on the Greek words, the main root word being encephalon, which means brain, or en meaning inside, cephalon means head. The prefix to each generally corresponds to its position along the length of the developing nervous system. The prosencephalon, or pros, which means in front, is the forwardmost vesicle, and the term can be loosely translated to mean forebrain. The mesencephalon, or mes meaning middle, is the next vesicle, which can be called the midbrain. The 3rd vesicle at this stage is the rhombencephalon. The first part of the word is also the root of the word rhombus, which is the geometrical feature with 4 sides of equal length. A square is a rhombus with 90 degrees angles, whereas prosencephalon and mesencephalon translate into the English word forebrain and midbrain. There is not a word for 4-sided figure brain. However, the 3rd vesicle can be called the hindbrain. One way of thinking about how the brain is arranged is to use these 3 regions: forebrain, midbrain, and hindbrain, which are based on the primary vesicle stage of development. The brain continues to develop and the vesicles differentiate further. The 3 primary vesicles become 5 secondary vesicles, and the prosencephalon enlarges into 2 new vesicles called the telencephalon and the diencephalon. The telencephalon will become the cerebrum, and the diencephalon gives rise to the several adult structures. Two that will be important are the thalamus and the hypothalamus. In the embryonic diencephalon, a structure known as the eye cup develops, which will eventually become the retina, and the nervous tissue of the eye called the retina. And this is a rare example of nervous tissue developing as part of the CNS structures in the embryo, but becoming a peripheral structure in the fully formed nervous system. The mesencephalon does not differentiate into any finer divisions. The midbrain is an established region of the brain at the primary vesicle stage of development and remains that way. The rest of the brain develops around it and constitutes a large percentage of the mass of the brain. Dividing the midbrain into forebrain, midbrain and hindbrain is useful in considering its developmental pattern, but the midbrain is a small portion of the entire brain, relatively speaking. On the other hand, the rhombencephalon develops into metencephalon and myelencephalon. The metencephalon corresponds to the adult structure known as the pons and also gives rise to the cerebellum. The cerebellum, which is from the Latin that means little brain, accounts for about 10% of the mass of the brain and is an important structure in itself. The most significant connection between the cerebellum and the rest of the brain is at the pons, because the pons and cerebellum develops out of the same vesicle. The myelencephalon corresponds to the adult structure known as the medulla oblongata. The structures that come from the mesencephalon and rhombencephalon, except for the cerebellum, are collectively considered the brain stem, which specifically includes the midbrain, pons, and medulla. So while the brain is developing, the anterior neural tube, the spinal cord, is developing from posterior neural tube. However, its structure does not differ from the basic layout of the neural tube. It is a long straight cord with a small hollow space down the center, and the neural tube is defined in terms of its anterior versus posterior portions, but it also has a dorsal ventral dimension. And as the neural tube separates from the rest of the ectoderm, the side closest to the surface is dorsal and the deeper side is ventral. And as the spinal cord develops, the cells making up the wall of the neural tube proliferate and differentiate into the neurons and ganglia of the spinal cord. The dorsal tissue will be associated with sensory functions, and the ventral tissues will be associated with motor functions. Early formation of the nervous system depends on the formation of the neural tube. A groove forms along the dorsal surface of the embryo, which becomes deeper until its edges meet and close off to form the tube. If this fails to happen, especially in the posterior region where the spinal cord forms, a developmental defect called spina bifida occurs. The closing of the neural tube is important for more than just the proper formation of the nervous system. The surrounding tissues are dependent on the correct development of the tube, and the connective tissue surrounding the CNS or the central nervous system can be involved as well. And there are three classes of this disorder, which are occulta, meningocele, and myelomeningocele. The first type is the spina bifida occulta. It is the mildest because the vertebral bones do not fully surround the spinal cord, but the spinal cord itself is not affected. No functional differences may be noticed, which is what the word occulta means. It is hidden spina bifida. The other two types both involved in the formation of a cyst, which is a fluid-filled sac of the connective tissues that cover the spinal cord called the meninges. Meningocele means the meninges protrude through the spinal column, but nerves may not be involved and few symptoms are present, though complications may arise later in life. Myelomeningocele means the meninges protrude and spinal nerves are involved, and therefore severe neurological symptoms can be present. Often surgery to close the opening of or to remove the cyst is necessary. The earlier that surgery can be performed, the better the chances of controlling or limiting further damage or infection at the opening. For many children with meningocele, surgery will alleviate the pain, although they may experience some functional loss because the myelomeningocele form of spina bifida involves more extensive damage to the nervous tissue. Neurological damage may persist, but symptoms can often be handled. Complications of the spinal cord may present later in life, but overall, life expectancy is not reduced. So this ends my lecture about the embryologic perspective of the nervous system and some disorders of the nervous system. And if you have any question, please write it in the comment section down below and I'll be glad to answer them all. And if you're still not subscribed to my channel, please also do consider subscribing. Hit the notification bell so that you'll be updated with my new videos. And also, if you like this video, please share and like. Once again, I would like to thank you all for watching my video and for listening to my discussion. So until my next video, bye everyone!